Hello everyone, thank you for coming today. In this occasion, I'm introducing MITNANEX, which is a new initiative for establishing a workflow for the processing of mitochondrial genomes from the whole genome sequencing with Oxford Nanopore data. This work is being developed with the Alianza Genómica de Medellín, which is made up of la Universidad de Antioquia con los centros de secuenciación UNICEX, el laboratorio One Health de la Universidad Nacional, Axomics de la Universidad de API y la Fundación Universidad de Antioquia. Maybe as some of you know, the mitogenome is very important from phylogenetics to research in disease and aging studies. And recently, with the increase in the precision of Oxford Nanopore technology and the relatively low cost for whole genome sequencing, it's an alternative for whole genome sequencing research. Several seed-free and seed-based pipelines for extracting the mitogenome from the whole genome sequencing have been developed, such as Mito, Novoplasty, Mito being Mingas, among others. However, these pipelines are only available for short reads, dominated by Illumina. In 2022, Mito Hi-Fi was the first pipeline for the extraction of the mitochondrial genome, but it only works with Bio Hi-Fi reads, and it requires a reference mitogenome. So to our knowledge, there was not an available pipeline for the extraction and annotation using reference and de novo the mitogenome from the whole genome sequencing using Oxford Nanopore. So in this occasion, I introduced MITNANEX, which is a flexible command line pipeline being developed that support the extraction and assembly of mitogenome from humans and other species with and without a reference mitogenome. So far, MITNANEX has offers two modes, a reference and a de novo mode. The reference, as its name suggests, it needs a reference mitogenome to extract and assemble the mitogenome, and also it produces variant annotations that I will talk about later. The de novo approach is a new proposal, so it still has some challenges, but it's intended to be a reference-free, and using a chimera approach, it can extract and assemble mitochondrial genomes. And this is a picture of how it looks on the terminal. So the reference mode workflow consists of 4 simple steps. The first step is to filter and map the reads to the mitochondrial reference genome. So far, it's only available the European form of the mitogenome, which is the RCRS. So here we use the most tested and popular tools for the processing of nanopore data. First, we filter the data with Chopper and map with Minimap. In this way, we can improve the alignment performance and remove some of bad qualities and possible nonce in the whole genome sequencing. After that, we do de novo assembly with Fly on its metagenomic mode in order to remove more bad quality and possible nonce and generate a consensus mitogenome, which is annotated with lead off using the reference mitogenome annotation. Mitogenome annotation with once we have the reads, the mitochondrial reads, then we use JTAK in Mutec 2 to do the variant calling. Since its haplotype assemble, it's using graph is perfectly suitable for nanopore data. We just have to modify some of the parameters to improve the results, and then the results of that variant calling is filtered with filter Mutec, and that output is annotated with BCF tools and MitoMaster in order to annotate clinical variants, not reported variants, and locus, the location of where these variants are falling in the genome, the mitogenome. So at the end, we got some principal output, which consists of the conceptual assembly of the mitogenome, the annotation of the mitogenome, the variant column BCF file, and a TSV tabular data from Mitomaster that is very comprehensive, that it includes also the upload group classifications and the reports of the variants. Some of the plots that MITNANEX can generate includes the depth distribution along the mitogenome, the mean base quality. We can also see the types of these variants, number of substitutions, number of transitions, transversions. Well, the purpose of this of establishing this workflow is under the project of sequencing centenarians people. So far, the Alianza Genómica is generating genomic data for the longevity studies. So far, we have sequenced 2 individuals, a male of 101 years and a female of 105 years, and we were able to assemble and annotate the genome with previously reported variants associated with longevity. We also could classify the upload group under the upload group A2B, which is a common upload group for Latin and Mesoamerican. And we could be able to identify in a total of 59 not reported variants that will need to be explored further, and we can see through MITNANEX the locations of these variants. The novo mode, as I mentioned, is still in development. It has been tested on a few fungal datasets, which includes a new fungus, a fungi that I was working on my undergraduate thesis called Talaromyces santanderensis. We were able to retrieve 100 of coverage with more than 99% of identity. Just in summary, this mode workflow relies on all versus all overlap alignments produced by Minimap and a custom Python script cluster the reads and separates the mitochondrial reads using the 3 nucleotide composition and using Kamen's algorithm. So for the future, we would like to, of course, improve the testing dataset and improve the reference and the de novo mode, and we hope to be sequencing more individuals in order to increase the samples and the data, but we need money. So thank you for la Alianza Genómica de Medellín, Suministros Islas, for our report. If you would like to do a partnership or would like to give some money, you can contact us, please, at this email. Thank you.
Thank you, Juan. Any questions from the audience?
So I may have missed it, but variants are really important in the mitochondria of many different diseases. With oxygen nanopore, there's still an error rate. When you do the assembly, obviously you can sense the sequence, but if you've got low coverage, you may still have errors in that sequence. Do you have any indication in the output of the coverage so you can assert confidence when you're very recording?
Good question. Yeah, we modify so far. We just modified the support of reads for each variant, so we have at least a minimum of reads that is consistent with the support.
And the error rate of the nanopore is increasing over the years.
Yeah,
but it has.
Yes, it's increasing.
So it's still there.
It's not zero.
Yeah.
It's still there. It's much better than the beginning, but it's still very nice work. Any other question?
Hello. Do you consider adding this apply to Galaxy?
In the future, yeah, it could be great. I haven't done that in my life, so I don't know. I don't work with Galaxy, so yeah, I do all in command line. So
Okay.
This is a good idea because you can have more awareness for people using more and more situations. So that's a great